Thanks everyone for joining us today for our webinar. Before we get started, I would like to introduce our speakers today. Carrie Horton is a genetic counselor in our cancer reporting group here at Ambry. Before joining Ambry, she was a clinical cancer genetic counselor for several years at Baptist Memorial Hospital and subsequently the West Clinic. Michelle Jackson is a genetic counselor and product manager here at Ambry Genetics, focusing on our hereditary breast and ovarian cancer and endocrine cancer tests. Prior to joining Ambry, she was a genetic counselor for several years at MD Anderson. And I am Karin Espenscheid. I am also a genetic counselor and product manager here at Ambry, focusing on hereditary gastrointestinal cancers and other cancers. Prior to joining Ambry, I was a genetic counselor for several years at City of Hope. So as many of you are aware, we have recently added several genes to our cancer panels, and today we wanted to just go over some of the literature related to each of those genes and what led us to adding each of these genes to the various panels. I will go over POL-E, POL-D1, and GREM1. After that, Carrie will cover BAF1 and SMARCA4. And then finally, Michelle will go over some of the recent data on FH and MEN1. Just to give you an overview, these are each of the panels that have had new genes added to them and each of the genes added to each panel. So to dive into POL-E and POL-D1, these two genes have been added to ColonNext, CancerNext, and CancerNext Expanded. Both of these genes are DNA polymerases, and they participate in DNA proofreading and replication. POL-E encodes the catalytic subunit of DNA polymerase alpha and is a leading strand polymerase, while POL-D1 encodes the catalytic and proofreading subunit of polymerase delta and is a lagging strand polymerase. The syndrome associated with mutations in these two genes has been called polymerase proofreading associated polyposis by some, but has not been adopted by all at this time. The syndrome is inherited in an autosomal dominant manner, and so far, most of the data published is on two specific missense mutations, which are in the exonuclease domain of these two genes. And we'll talk more about those. So there have been two key publications that have the strongest data on these two genes. The first is by Pallas et al., and the second is by Valley et al. So Pallas et al. Did whole genome sequencing of 13 families who had been previously tested for the common hereditary colorectal cancer syndromes, Lynch syndrome, FAP, and MAP, without mutation identified. And they did linkage analysis and association studies and identified two alterations among three families, one in POL-E and one in POL-D1. After that, they screened for these two specific mutations in large cohorts of cases and controls. And they identified 12 cases with the POL-E mutation and no controls, and one case with the POL-D1 mutation and no controls. Some of the analysis and assessments that they did for the POL-E mutation to determine that it was in fact a mutation included the fact that it mapped to the active site of the exonuclease domain, and prior studies on somatic mutations in this gene identified that it causes a mutator phenotype in yeast. For the POL-D1 mutation, it's in a highly conserved region and is close to other mutations that also have a mutator phenotype in yeast. All of the tumors that were assessed in this study were microsatellite stable. However, I think it's important to point out that most of these tumors were not carcinomas. Most of them were actually adenomas, and I think that's important because we do know that even in Lynch syndrome tumors with microsatellite instability, we often cannot see that instability until the tumor develops into a carcinoma or at least has some high-grade dysplasia. In the initial three families that were identified with these mutations, all of the carriers were affected with either colorectal cancer or adenomas. and all of the affected individuals ended up being carriers. So there was perfect concordance. The Pol E family had colorectal cancer and adenomas only, while the two Pol D1 families had a combination of colorectal cancer, adenomas, and endometrial cancer. One of the individuals in one of the families also had a history of two astrocytomas, but he did have a clinical diagnosis of NF1, which was tracking from the opposite side of the family from which the Pol D1 mutation came. Palace et al. Summarized and concluded that the Pol E alteration predisposes to multiple colorectal cancers and adenomas, and that the Pol D1 alteration predisposes to colorectal tumors, endometrial cancer, and maybe brain tumors. They concluded that these mutations are highly penetrant, and also that they cause an increase of base substitutions, but not microsatellite instability. They also hypothesized that other mutations in the exonuclease domain of these two genes may also cause the same phenotype. The next paper by Valle et al. Screened 858 cases for the same previously identified two mutations. In 612 of those cases, they screened for just those two mutations, and then in 246 of those cases, they sequenced the entire exon that mutation is in. They identified one additional Pol E mutation carrier, which they determined to be de novo after testing the parents, and identified a new Pol D1 mutation due to the fact that the whole exon was sequenced in one of the cases. And this particular mutation is just a few base pairs down from the other mutation identified in Pol D1. This particular mutation, or in this case, the phenotype of the patient fits with the previously identified Pol D1 mutation carriers, and the co-segregation within the family also fit. It's a highly conserved residue in the proofreading domain of this gene, again, and had been shown to cause a mutator phenotype in yeast. Also, interestingly, this particular amino acid location is the paralog of Pol E L424, which is where this particular Pol E mutation has been identified. Some of the cases in this study did have MSI and IHC previously performed. However, it's unclear whether these particular cases where the mutations were identified had previous MSI and or IHC. So these are the two families. And you can see that the Pol E mutation carrier had over 31 adenomas and a couple of other polyps, and that although the father had colorectal cancer at 56, he did test negative for the mutation, as did the mother of the patient. And here is the Pol D1 family, colorectal cancer at 36, as well as a GIST. Some other gastrointestinal and endometrial cancers in the family, although the only other individual who was actually tested was the maternal aunt, who has a diagnosis of colorectal cancer and endometrial cancer. Here's another paper that I would like to review. In this case, they screened 1,188 cases, also had been previously tested for Lynch syndrome, FAP, and MAP, and in this particular paper, they also only screened for the two previously identified mutations. They also, in this case, did MSI and IHC for the mismatch repair proteins, and did IHC for Pol E, just to see if that was a useful screening tool. They also did tumor sequencing for the cases. So they identified 3 additional Pol E mutation carriers and were able to test a relative of one of those carriers. So the first case with a Pol E mutation had an MSI high colorectal cancer that also had loss of MSH6 in the tumor. Although this person did have a Pol E mutation, there was not loss of Pol E staining in the tumor. And when they looked at the tumor sequencing, there were many different alterations in the tumor, including 3 in MSH6, one of which was clearly truncating. They were able to test the son of this individual, and he also had an MSI-high tumor, which interestingly showed loss of both MSH2 and MSH6, although there was not loss of POL-E. So they concluded that POL-E IHC is not a useful screening tool for POL-E mutations. This individual had an MSH2 truncating mutation in his tumor and had a APC likely pathogenic alteration. The third individual had an MSI-high tumor, also with loss of MSH2 and MSH6, although the only somatic alteration that they saw was a probably not pathogenic alteration in MSH2. The final individual with a POL-E mutation identified in this paper had a microsatellite stable tumor, and apparently these studies were done prior to this particular publication, and the tumor was not still available for them to do somatic sequencing in this case. So in this study, they actually found POL-E mutations in individuals with microsatellite stable and microsatellite unstable tumors, and they concluded that POL-E mutations can be associated with a Lynch syndrome phenotype, both in the patient and in the tumor. And they hypothesized that the POL-E hypermutator phenotype may cause all of these somatic mutations, some of which may occur in the mismatch repair genes. So most of the mutations reported so far are within the exonuclease domain of the two genes, and primarily two specific missense mutations. Here is the only mutation reported that I could find outside of the exonuclease domain, but one has been reported, and it is this deletion of 2 base pairs in an individual with colorectal cancer at 26. Another POLD1 phenotype has been reported, and that is it's a pretty unique phenotype associated with a de novo single amino acid deletion. This particular deletion happens to occur in the polymerase active site, not the exonuclease domain, and it's been found in 4 probands with essentially a premature aging phenotype. So the overall clinical phenotype for mutations in POL-E and POLD1 is primarily early onset colorectal cancer and adenomatous polyps. The summary of the published information related to polyps is that the count of polyps can range from anywhere to 2 to over 100 polyps, and the age of diagnosis has ranged anywhere from 60 to 74. For colorectal cancer, the range of ages of diagnosis in the published cases ranges from 22 to 78, although the average age is 40, so they do tend to be early onset. And the other cancers reported in these families include ureter cancer, endometrial cancer, brain, ovarian, GIST, and duodenal cancer. So over the last several years, the data has accumulated related to these 2 genes. They suggest these two genes to be highly penetrant, and identifying individuals with increased risk for colorectal cancer allows for screening, prevention, and early detection. So, due to that fact, and obviously many requests from many of you, we felt it was time to include these 2 genes on our panels. You may be wondering why we did not choose to add them to Ovanext at this time. We felt that the data on the gynecologic cancer risk associated with these 2 genes is more limited than the colorectal cancer and polyposis data, so we decided to hold off on including them in Ovanext until more data accumulates on that particular cancer risk. Moving on to GREM1. GREM1 has also been added to ColoNext, CancerNext, and CancerNext Expanded. It is also known as gremlin-1 and is a bone morphogenic protein antagonist, so not a tumor suppressor, not involved in DNA repair like many of the other cancer predisposition genes. It is involved in organogenesis, body patterning, and tissue differentiation. And the syndrome it's been associated with is called hereditary mixed polyposis syndrome. So far, only one mutation has been seen associated with this syndrome, and it's a 40 kb duplication upstream of GREM1. So far, all of the cases reported are of Ashkenazi Jewish ancestry, and this particular duplication is also inherited in an autosomal dominant manner. So there is one key publication that really is the only one to have demonstrated this duplication in association with hereditary mixed polyposis syndrome. It's the Jagger et al. Paper, and they had been following several large Ashkenazi Jewish families with hereditary mixed polyposis syndrome and had identified a region of interest by linkage analysis. However, they hadn't been able to find any sequence-based mutations in that region, but after doing an array, identified this large duplication in 2 cases. The duplication is a tandem tailhead duplication that also includes an insertion of a 30 base pair sequence of DNA of unknown origin. The large duplication ranges from intron 2 of the previous gene, SCG5, to just upstream of GREM1. They found increased levels of the GREM1 transcript in normal tissues of the cases who carried this duplication, but did not find a difference in the expression of SCG5. So this led them to determine that this duplication leads to upregulation of GREM1. They then tested 40 affected and 50 unaffected individuals from 6 families with hereditary mixed polyposis syndrome, and there was perfect concordance, meaning that all of the people with mixed polyposis had the duplication and the healthy individuals did not. And these are the 2 families, or 2 of the families, and you can see there's a mixture of polyp types, adenomatous and hyperplastic here, also in this family. So we decided to add this gene to our cancer panels, again, because of an accumulation of data. Although there is only one key publication so far associated with this duplication, is a strong publication with multiple families, perfect concordance between the mutation and the disease status, and the data suggests the mutation to be highly penetrant. With that, I will turn it over to Carrie.
Hi, everyone. So our next gene to discuss is BAP1. This stands for BRCA-associated protein 1, which codes for a deubiquitinating enzyme. It's involved in cell cycle and gene expression regulation, signal transduction, protein trafficking, and double-stranded DNA repair. It's actually recruited to sites of double-strand DNA breaks and participates in homologous recombination with BRCA1. And preliminary data so far suggests that it appears to confer susceptibility to synthetic lethality from PARP inhibitors, much as the BRCA mutations would. So for BAP1, there's far more publications than the other 3 genes that have just been presented. So rather than try to provide an exhaustive review or focus on individual phenotypes, I thought I'd touch on some of the findings from several studies on different tumor cohorts that kind of came together at the same time. So in 2011, Weisner et al. Took a look at kindreds of melanocytic tumors and performed array CGH in 22 tumors from individuals in 2 families and found a frequent loss at 3p21. So they went ahead and sequenced the minimally deleted region in the germline of 6 affected individuals, which identified BAP1 mutations. There was one truncating mutation in one family, and the other family had a splicing mutation. This segregated with disease in 8 individuals, and the authors noted that they observed uveal melanoma in one family and cutaneous melanoma in the other. Also in 2011, Testa et al. Was studying mesothelioma kindreds. They had a similar process of linkage and tumor evaluation, which led to the identification of BAP1 truncating and splicing mutations in 2 familial mesothelioma kindreds, as well as in 2 of 26 sporadic mesothelioma patients. But importantly, both of the sporadic positive mesothelioma patients actually had a personal history of uveal melanoma, and none of the 24 negative sporadic patients did. We also have a cohort of uveal melanoma kindreds, and at this point in time, kind of putting together the pieces, these researchers went straight for direct BAP1 sequencing in 53 individuals with uveal melanoma that did not have CDKN2A or CDK4 mutations, and found a truncating mutation in one individual. And in this family, it did segregate with uveal melanoma, cutaneous melanoma, mesothelioma, and one case of meningioma in the family. They also identified the mutation in one unaffected 55-year-old. So later on, Popova et al. Started looking at renal cell carcinoma kindreds. In one family in particular, they tested their proband. They did whole exome sequencing in their proband, who had 3 primary renal cell carcinomas in her 30s, and found a BAP1 mutation, which they confirmed in her paternal cousin, who had a renal cell carcinoma in his 40s, which made both the father and the paternal aunt with renal cell carcinoma obligate carriers. They went on to show, though, that 0 of 32 VHL negative renal cell carcinoma families had mutations in BAP1, prompting them to suggest that it's actually a rare cause of renal cell-only families. In 11 of 60 patients from families with uveal cutaneous melanoma or mesothelioma with or without renal cell carcinoma, 11 of 60 of those had BAP1 mutations, so that's about 18%. And at this point, they actually had enough families to calculate a risk estimate for renal cell carcinoma, and using a lifetime risk of 0.5% in the French population for renal cell carcinoma, they showed that there's a 15-fold increased risk for males and a 59-fold increased risk for females. And it could be that this range in between males and females could be because of a gender-specific incidence of the disease itself. It could be that this is still a small sample size that they're drawing from, or it could be a little bit of both. So in 2014, Polarski et al. Put out a nice paper that had actually a couple pedigrees that kind of illustrate and capture more of the gestalt of what these BAP1 families look like. Here we have a family with uveal melanoma, mesothelioma, and renal cell carcinoma. Here we have cutaneous melanoma and mesothelioma, and here's a family with all 4 of the core BAP1 cancers. This article also includes proposed management options, in addition to, of course, good practices of follow-up with a primary care physician and adherence to American Cancer Society guidelines. They also suggest annual ophthalmological exam starting at age 11, which is 5 years younger than the earliest reported uveal melanoma patient, and referral for patients with pigmented lesions to an ocular oncologist for follow-up. They also suggest annual derm exam starting at age 22, which again is 5 years younger than the earliest reported cutaneous melanoma. And I thought this would be nice to include something to refer patients to in the event of positive results. So overall, what we know right now, the clinical phenotype of BAP1 mutation families includes increased risk for renal cell carcinoma and uveal melanoma, which includes melanomas of the iris, ciliary body, and choroid. This may prove to extend to ocular melanoma, which includes as well conjunctival melanoma. At this time, I didn't see any patients described having conjunctival melanoma, and it could be that that's because it actually only makes up 10% of ocular melanoma, so we'll see if that expands. In addition, there's an increased risk for cutaneous melanoma, asbestos-induced mesothelioma, and a specific type of non-malignant tumor called non-malignant melanocytic BAP1 mutated atypical intradermal tumors. So This is kind of a newer designation, and by definition, it involves molecular characterization. You might not actually see this on path reports. It may be that these are just described as melanocytic tumors, or perhaps Spitz-like melanocytic tumors, which these bear close resemblance to. And in the published literature so far, individuals have anywhere between 5 and 50 tumors, presenting most often in their 30s to 70s. Rationale for addition to a panel. As always, we take a lot of cues from our clinicians and helping to look through the literature and point out our attention to new studies that have come out. And there really have been several publications of numerous families with overlapping phenotypes, suggesting highly penetrant phenotype. And I think it's important, too, that with this gene, a number of the associated tumor types have good surveillance options available. And in addition, uniquely, I think information about this mutation could actually influence vocational or housing decisions to avoid asbestos exposure at all costs. Okay, so moving on to SMARCA4, which is going on OvaNext, CancerNext, and CancerNext Expanded. This stands for SWI SNF-related matrix-associated actin-dependent regulator of chromatin, subfamily A, member 4. So it encodes the catalytic subunit of the SWI SNF chromatin remodeling complex. So basically, it's a chromatin remodeling gene that helps to regulate cell cycle progression, cell death, differentiation, genomic instability, and DNA repair. Inactivation of multiple genes in this complex have been reported in tumor types such as colon, lung, and classically rhabdoid tumors. What you may see in some of the somatic or tumor literature is that this is often referred to as BRG1 in that context. One article when I was going through the literature really kind of grabbed me and made me think about the evolution of our understanding of this gene and the associated tumors. So I thought I'd kind of walk through this group's experience as a way to introduce the supporting literature. Witkowski et al. In 2013 put out an update on a case that they first published in 1993 of a 27-year-old female with immature teratoma of the ovary discovered during pregnancy, whose daughter was later found to have an immature teratoma of the brain, first observed in utero. They were TP53 negative, and there was really nothing else to offer them at that time. The daughter passed away in infancy. 20 years later, this must be just one of those cases that stuck with the group, and they revisited the case. They performed exome sequencing on DNA samples that they had banked from the mother and daughter, and also went ahead and exomed 106 germ cell tumors, since germ cell tumors are related to immature teratomas. They found identical SMARCA4 germline mutations in the mother and daughter, but not in any of the other germ cell tumors. So meanwhile, while this is kind of going on, 2 other independent studies identify SMARCA4 mutations in 3 unrelated individuals with rhabdoid tumors. In 2010, Schneppenheim et al. Described ATRT, atypical teratoma rhabdoid tumor, in an 8-month-old, and a rhabdoid tumor of the kidney in her 7-month-old sibling. SMARCB1 testing was performed first because that's the gene that more typically is associated with these tumor types. That was negative, so they did testing on the other SWI SNF genes and found identical SMARCA4 truncating mutation in both sibs, as well as, interestingly, the unaffected father. Now, despite the unaffected father, authors argued that this alteration is the cause of disease based on the rhabdoid tumor of both siblings lacking expression of SMARCA4 protein, the fact that the alteration was identical in both sibs, the fact that RT-PCR on lymphoblastoid cells and expression studies indicated that nonsense-mediated decay was the molecular mechanism for the lack of expression. due to the fact that copy-neutral loss of heterozygosity at the SMARCA4 locus was identified as a second hit in the tumor cells, and the fact that the other classical genes, including SMARCB1 and 3 additional genes of this complex, did not show any alterations. Hasselblatt et al. Describe a 9-month-old boy with an atypical teratoid rhabdoid tumor demonstrating intact SMARCB1, which is not what they would expect, and absent SMARCA4. And the tumor was shown to be homozygous for a SMARCA4 truncating mutation. In this case, both parents refused germline testing for the patient and themselves. But it was kind of enough for Witkowski to kind of look back and say, let's look at the path again on these patients. So they initiated path review of both the ovarian and brain teratomas, and they actually were able to reclassify them as malignant rhabdoid tumors, an atypical teratoid rhabdoid for the daughter and a small cell carcinoma of the ovary, hypercalcemic type for the proband. So I just thought that was just amazing to think of the years that they were putting this together and thinking, well, why would our patient have the SMARCA4 mutation and none of the other germ cell tumors have it, and kind of waiting until everything had to add up in order for them to have a light bulb moment and reclassify the path. So other studies went on to corroborate findings of SMARCA4 germline mutations in individuals with small cell carcinoma of the ovary, hypercalcemic type. So a little bit more on that. It's a highly aggressive tumor with age of diagnosis ranging from 9 years to 43 years in the published literature, median age being 27. Survival ranges from 5 months to several years for stage 1 and 2 months to 23 months for stage 3. So it's a real bad actor. In addition to morphologic features, small cell carcinoma of the ovary is also characterized by increased serum calcium levels. And more recently, this has been reclassified as a true malignant rhabdoid tumor of the ovaries in 2014 by Folks et al., and the authors call for it to be renamed as such. So our clinical phenotype for this gene looks like malignant rhabdoid tumor of the ovary, which you may say interchangeably with small cell carcinoma of the ovary. Malignant rhabdoid tumor of the CNS, which is designated atypical teratoid rhabdoid tumor. And I think the verdict is still out on the malignant rhabdoid tumor of the kidney. There's only been one case report so far, so we'll see what happens there. Unfortunately, tumor risks are unknown at this point. It seems to demonstrate extremely variable expression and penetrance, with some individuals presenting in utero and others being into adulthood and unaffected. I kind of think about it a little bit like TP53, where sometimes you have infants with choroid plexus or adrenocortical carcinoma, and then others who are in their 20s or 30s before a breast cancer, or even later onset 30s and 40s with, a glio or something like that. So I kind of think of that similarly, but certainly one that deserves special attention and counseling. A rationale for adding it to the panel, honestly, it's a very rare, very distinct tumor type, so why would we include it? I think the answer to that would be that this tumor type is exceedingly difficult to characterize. I think it's the story of Witkowski 2013 could be seen again, where you're working with a patient who has, maybe high-grade undifferentiated carcinoma, and it turns out that this result actually helps characterize their true diagnosis. So I included some images from a later Witkowski article just listing the numerous tumor types that this that small cell carcinoma of the ovary hypercalcemic type mimics. So ultimately, identifying mutations will have a significant implication in accurate diagnosis of the tumor itself, as well as prognosis and familial risks. So now we'll move on to Michelle's segment.
Hi there, everyone. So I'm going to switch gears just a little bit, just because the two genes that I will be presenting about, we already know lots of information about them historically. And then also talking about the information that we've used, the data, to add these to the PGL Next panel, and for MEN1 to add to Cancer Next Expanded as well. So first I will discuss the FH gene, which will be added to PGL Next. The FH gene is the fumarate hydratase gene located on chromosome 1q42.1. It has 10 coding exons and encodes 510 amino acid protein. It's involved in Krebs cycle catalysis and tumor suppression. I'm not going to go into great detail about the Krebs cycle, but really just wanted to show this picture as an illustration for previous genes, the SDH complex of genes that we're aware of that cause hereditary paragangliomas and pheochromocytomas, and just to show that the FH gene is just downstream of that in the Krebs cycle, to kind of make that connection of the plausibility of paragangliomas and pheochromocytomas to be associated with mutations in this gene. So we're pretty aware of the clinical phenotypes that can be associated with FH gene mutations. In autosomal dominant fashion, we're aware of HLRCC, or hereditary leiomyomatosis renal cell carcinoma, which has increased risks for cutaneous and uterine leiomyomas. These are typically benign but may have malignant potential. And there have been case reports discussing the malignant potential of those tumors. Also, renal tumors, up to a 20% lifetime risk for those. Most of those are classified as type 2 papillary renal cancers. There have been other pathologic types of renal tumors seen with HLRCC, but most of them do tend to be pretty aggressive and can be diagnosed even in childhood. And then kind of this new addition of paragangliomas and pheochromocytomas, which I will talk about in the next few slides. In the autosomal recessive mode of inheritance, we're aware of fumarate hydratase deficiency, which has a very different and very severe phenotype that can occur, including severe developmental delay, infantile encephalopathy, which includes severe seizures and dysmorphic facial features. So in focusing on the autosomal dominant mode of inheritance for FH and the literature that has been published in 2014 regarding its association with paragangliomas and pheochromocytomas, I will review these two articles. So the first article, Castro Vega et al., looked at 598 unrelated patients from France and Spain who had been diagnosed with either PHEOs, which I'm noting here as PCC, and or paragangliomas, which I'm noting here as PGL. In all of these individuals, they looked for mutations in all of these genes listed, which happens to be the previous version of our PGL Next panel, which no mutations were identified in any of these genes for this subset of patients. They had an initial patient that had an SDH-like molecular phenotype and was negative for all of for mutations in any of these genes. Therefore, they proceeded with whole exome sequencing and identified FH as a candidate gene mutation for that particular patient. For the rest of the group of patients, they proceeded with finger sequencing and MLPA to look for any mutations in the FH gene. In addition, they also did somatic testing. They looked at loss of heterozygosity, IHC, staining, and enzyme activity. For their germline results, they identified 41 variants in the FH gene. They ruled out several variants that were SNPs with general population incidence, the missense variants, and things of that nature. But they did ultimately identify 5 pathogenic mutations, at least that they classified as pathogenic. For the somatic testing, of those 5 that were classified as pathogenic, they had 3 tumor samples available, and you can see below in this table those 5 individuals that were identified. For the somatic testing, all demonstrated loss of heterozygosity. There was also a correlation of low 5-HMC, which is hydroxymethylcytosine, and positive immunostaining for 2-SC, which is 2-succinylcytosine. With all of these correlations together in the tumor and the mutations that they identified, they determined that these 5 FH mutations were pathogenic and conferred an increased risk for paragangliomas and pheochromocytomas. Interestingly, as well, several of these individuals, 3 of the 5, were found to have metastatic paragangliomas or pheochromocytomas, and the age ranges varied as well, anywhere from 20 to 70 years of age in this population. So the authors stated that with the Krebs cycle enzymes being increasingly reported to play a role in predisposition to inherited tumor syndrome, they felt like FH really should be considered as a predisposition to pheochromocytomas and paragangliomas. There have been other reports of other genes in the Krebs cycle possibly conferring increased risk for pheos and paragangliomas as well. They also estimated that germline FH mutations accounted for approximately 0.83% of cases, which was similar to TMM127 and the MAX genes, which are on several panels and known to cause pheos and paragangliomas as well. So they felt that with all this information together, it really made a strong case for them. So moving on to the Clark et al. Study, following the Castro-Vega et al. Study, they had 72 patients in their study that had a history of either pheochromocytoma, paraganglioma, or a head and neck paraganglioma. So they differentiated between those 2 groups. Their initial patient was a pediatric patient who was diagnosed with a pheochromocytoma, for which they did whole exome sequencing and identified an FH mutation and no mutations in any of the other genes. Therefore, they went on to review the data from 21 additional patients. Germline, they did Sanger sequencing of all 10 exons of the FH gene. And then for somatic testing, they also did some functional analysis. So combining their results with the Castro-Vega et al. Study, they actually increased the incidence of FH mutations in pheos and paragangliomas to 1.045. Interestingly enough, neither of their patients, so they identified 2 additional patients to the Castro-Vega et al. Study. Neither of those patients had malignant tumors, although they did state that their follow-up was less than 5 years, and they did identify a pediatric case as well. So you can see in this summary of the 2 groups, the 7 patients that have been identified total, their clinical phenotype anywhere from 6 years of age to 70 years of age, so a very wide range of diagnosis. And then also the varying distinction between benign and malignant pheos or paragangliomas, which I think is important when considering testing these individuals. This is just a summary slide of all of the FH gene mutations that have been identified previously. The notations in red, which I apologize, it's a little blurry, so it is hard to read, but you can see the red mutations noted are the ones identified in these 7 patients, and they are throughout the gene. There's not any sort of particular hotspot or region of the gene that seems to favor an increased risk for pheos or paragangliomas. So these authors concluded that FH should be included in gene panels for hereditary or for inherited pheos and paragangliomas, including head and neck paragangliomas. And then it's possibly that pheos and paragangliomas might develop in patients with HLRCC, and that it is unclear whether individuals with FH-related pheos or paragangliomas are at higher risk of developing features of HLRCC, so going both ways. And as the last slide demonstrated with the mutations throughout the gene, it's really difficult to determine what the overlap may be or if all individuals with FH mutations will have an increased risk for both conditions. They also noted that long-term follow-up of both of these groups will be very crucial to determine what tumors
or features these individuals may develop and what the best management should be for these individuals. So our rationale for adding the FH gene to the PGL-Next panel, are really these two studies that identified multiple probands, included the supporting tumor analysis that really showed a strong correlation of the FH gene with PHOs and paragangliomas. The data suggesting malignant potential, which will really be imperative for proper follow-up management of these patients, and particularly the young patients presenting with PHOs or paragangliomas. They may not have any other features that could indicate any particular gene mutation, and therefore a panel would be very appropriate for those patients. So I'll move on to the MEN1 gene. It's being added to PGL-Next and Cancer-Next Expanded. Again, we're all very familiar with MEN1, the menin1 gene on chromosome 11q13.1, 610 amino acid protein. It acts as a tumor suppressor gene through its involvement in transcription regulation, cell proliferation, apoptosis, and genome stability. Again, the clinical phenotype has been pretty well established for MEN1, autosomal dominant. It can either cause MEN1 or familial isolated hyperparathyroidism, or FIHP, which is really just characterized by primary hyperparathyroidism as the sole feature in a family. However, with MEN1, it's characterized by multiple neuroendocrine tumors. Primary hyperparathyroidism is obviously the most common. GEP, neuroendocrine tumors, pituitary adenomas, adrenocortical tumors, which if you look back, pheochromocytomas have been included as a rare feature of MEN1 for quite some time. Bronchial and thymic carcinoids, and then additional dermatologic features as well. In historical studies, most patients should exhibit some sort of clinical or biochemical symptoms by age 50 if they have MEN1. So in thinking about the literature review for MEN1, I wanted to include the gene reviews because I think they've included pheochromocytomas more in the most recent edition of the gene reviews for MEN1, and so I think that adds to our rationale. But then also there was a very recent paper that came out that I'll discuss further in the next few slides. So the Denise et al. Paper looked at 39 patients with PHOs or paragangliomas and pituitary adenomas in a sporadic or familial setting. In the familial setting, they classified the familial part of it as 2 or more subjects with pituitary adenomas without syndromic features of other diseases such as MEN1 or Carney complex. They also ruled out NF1 based on clinical criteria according to published guidelines. For the controls, they used probands from 23 probands who were negative for AIP mutations, which has been shown to cause familial isolated pituitary adenomas. They looked at several genes to rule those out in this patient population. They used finger sequencing and MLPA, and then also did somatic testing as well, loss of heterozygosity and IHC. So their results, they found 19 germline mutations in patients with PHOs or paragangliomas and or pituitary adenomas. These are the genes listed that they identified mutations in. And 20 of their 39 patients had no identifiable mutations in any of the genes tested, 10 of those having both PHOs or paragangliomas and pituitary adenomas. And then no mutations that were identified in the AIP gene or CDKN1B. So just looking at this table a little bit more closely, I really focused on highlighting the FDHV gene and the MEN1 gene. This is really where they were able to look at the tumor a little bit more closely in these cases. So the FDHV gene, they identified 9 patients with variants. 8 were classified as mutations. 1 was still classified as a variant. 3 tumors were available. 3 pituitary adenoma tumors were available that demonstrated loss of heterozygosity. And then one pheochromocytoma was available that also demonstrated loss of heterozygosity. For MEN1, there were two mutations identified. The pituitary adenomas were not available for tumor testing, but the pheos were, and both of those demonstrated loss of heterozygosity as well. So really the author's main conclusion was genetic testing for germline mutations in FDH complex genes and MEN1 should be considered in patients with a constellation of pheos or paragangliomas and pituitary adenomas. So for our rationale and adding MEN1 to PGL Next and Cancer Next Expanded, these historic multiple case reports of MEN1 kindreds with pheochromocytomas. In addition to the Denny's et al. Study, patients with a pheochromocytoma and one other MEN1 type of tumor or possible MEN1 feature. Thinking about, again, young patients presenting with pheochromocytomas and maybe not demonstrating any other features, could also have MEN1 mutations that may not be identified otherwise without a panel. And then really thinking about pheochromocytomas in the context of MEN1, and that historically we haven't really looked for those in a more global way. And so there may be an underestimation of pheochromocytomas in MEN1 and as a feature of MEN1 as well. So these are a list of our references that we discussed today.
Thanks, Michelle. So I want to thank everyone for joining us today. The next educational webinar that we will be having will be a CEU webinar related to hereditary cancer. It's on Tuesday, June 2nd at 10 a.m. Pacific time. It is called Cross-Border Conversation, Clinical Approaches to Hereditary Cancer Panel Use. It will be presented by Jenna Scott and Christine Kobelka, both clinical cancer genetic counselors. So please feel free to visit our website to register for that webinar. We hope to see you then.